Principal Vice-Chancellor, thank you for your kind introduction, and thank you, everybody, friends and colleagues and members of the public, for coming this evening. It's a wild and windy night. So it's Christmas. And I'd like you to transport yourselves in your mind to Christmas Eve. And you're out in the Scottish countryside with family or friends. And the fire's roaring, the wood-burning stove, Grandad's snoring away in a comfy sofa, half glass of whiskey beside him. The grandchildren are playing under the tree. It's a very modern Scottish family. So Mum is sitting beside the fire, reading her favorite magazine, and Dad is in the kitchen slaving over the turkey for tomorrow. And then, in the darkness, you hear a sound. The unmistakable sound of the barn owl. And the barn owl wants his Christmas supper too, just like you do. And so he flies down from a perch in the trees onto some unsuspecting little vole or mouse who's scurrying around in the snow underneath and catches him. So the question arises of how does the barn owl do that? Well, he uses his brain, of course. But how does he use his brain? Well, this problem has been studied very intensively by a great scientist called Eric Knudsen at the California Institute of Technology. And he worked out that there was very small time delays between the sounds reaching the left ear and the right ear in the barn owl, and that the nervous system was able to detect that difference in timing, the interaural time delay. And on top of that, the barn owl ears are at slightly different positions on the left and right, so there's actually a vertical difference as well. And what the barn owl does is it feeds through neural impulses through a circuit into a region called the lemniscus. And the lemniscus has the following interesting architecture, which is a series of delay lines. Sorry, this doesn't work, but it's okay. In which information's coming from the left side and information's coming from the right side. And I think you can see in the panel on the left that the time it would take for the nerve impulses to get to neuron number E would be longer than to get to neuron number A, and vice versa for the sound coming from the other side. So what this little circuit does is it converts a time delay into a place code. And once you've done that through this little neural circuit, then you're able to arrange that the firing of cell A shifts the position of the head of the barn owl in one direction, whereas the firing of neuron number E shifts it in another direction. So this transformation of the time delay to a place code, thanks, John, does something really very magic. It not just solves the problem of catching the prey, but also transforms the representation of information in the brain. So in this example is the essence of the argument I want to put to you today, which is that brain cells, their chemical messengers, the receptors, the lock and key mechanisms that many study, and the molecular mechanisms inside them are all absolutely vital for function. But it's the layout of the anatomical circuit that's critical here to prey capture. So when we think about the human brain, we're often flooded by all manner of facts that are really a little hard to understand. 100 billion cells, up to 5,000 synaptic connections, and so on. all in 1.4 kilograms inside our head. It's difficult to make sense of all of these numbers. But the point I want to put to you is that inside this brain, and it's critical in my view to understanding memory, are networks that represent experience in different ways, and these networks can change in response to experience, and those changes in those networks are the basis of memory. Now, in the political world, they have a slogan. I think it was invented by Bill Clinton, but no doubt many others, which is, "It's the economy, stupid," that you only have to think about when it comes to an election. So my mantra here is, it's the neural circuit, stupid. And it's not a new idea at all, because Ramon Cajal, the great Spanish anatomist, thought much the same in his very detailed studies of the anatomy of the brain. Okay, so now let's take that argument and apply it to memory. I think we have to start by defining what memory is and why it matters. So, there are lots of different aspects of memory. There's the sort of incidental and automatic things that are just happening as you go about your daily life, remembering what you had for breakfast, or where or when some recent event happened, and what that event was. There's more deliberate types of memory, like conscious, effortful learning, acquiring a skill, such as riding a bicycle, learning a foreign language, as I'm struggling to do to learn Spanish. And then on top of that, there's memory time travel. The fact that what memory enables you to do is to move from the particular moment in time, both backwards and also forwards, to reminisce about the past, but also to envisage the future. So those are some basic features of memory, which we're all aware of, but perhaps it's valuable to be reminded of as we think about this particular problem. Now, one fascinating aspect of memory for me is remembrance, a kind of cultural act of memory in which we all engage. Now, I live in the southwest of Edinburgh, and so as I drive up the Collington Road every morning and every evening back to home, I drive past the Craiglockett Hospital here, and those who know the southwest of Edinburgh will know this place well. It's near the Redford and Dregford Ballads. And this hospital was built shortly before the First World War, and then during the First World War, W.H.R. Rivers persuaded the generals that instead of shooting the soldiers at dawn for cowardice, that he could treat them for shell shock. And in this hospital, there were all manner of treatments developed that could help people recover. And of course, amongst those was the famous war poets like Severy Sassoon and Wilfred Owen. And in the front entrance here is a little library which has many of the books of the war poets. And as we now, 100 years later, remember the First World War through plays like The War Horse and various other ways, then we have in remembering something that we never actually experienced. So it's not recall in the conventional sense, it's more a matter of learning new things. And that then takes on a further dimension where, as we saw in London with the poppy field around the Tower of London, it began as an idea to mark each man and woman who had died in the First World War. And as it built up, it caught the imagination first of Londoners and then of the whole nation, and people came to see this amazing spectacle. So I think that's an example of, if you like, the importance of memory, but it isn't necessarily something that happens every day. There's the memory of events, specific things like a couple's first meeting, and these constitute anchors, I think, in our life. There's all manner of shared activities, knowledge of each other's customs, and in this way memory connects brain and body, and I think it even reaches from one person to another. Now, there's a very interesting new field called embodied cognition. Indeed, the professor of philosophy in this university is a proponent of this view. And it has the theme of thinking about conscious and non-conscious aspects of predictability in our lives. And I think that memory is a somewhat forgotten aspect of this, but an important aspect of this. And then finally, there's the question of when things go wrong, in an accident, a personal crisis, worry about a relationship. And there, I think, not only emotional attachment and love, but memory serves collectively as a kind of glue that can help to hold things together. But on top of that, quite apart from shared memory, there's the externalization of memory that's happening in all of our lives. From Gutenberg onwards, we've had ways of sharing knowledge freely and easily through books. And then came the gramophone and the radio and television, through which various experts were able to give us examples of their music or radio programs or TV programs. But now, so much of human knowledge is sort of democratized in that we can all participate. And in a way, knowledge is just a mouse click away. Do we really need to remember phone numbers anymore when all we have to do is click on our smartphone? Do we need to remember how to find our way around when we're just using GPS systems? Where will this all go? I don't know. I'm sort of somewhat conservative about this and think it's important to keep these things going, but maybe it won't be too long before we have our emails on our gravestones so that we can remain connected even in the afterlife. Now, often we come to appreciate the value of something when it's lost. And I think memory is no exception to that rule. And so I think it's important to mention that there have been a number of cases of patients who've been studied very intensively and through their careful study have taught us a lot about memory. Perhaps the best known of this is patient HM, always known by his initials until he died, and now known as Henry Meliasen, and studied by Brenda Milner and also by the late Suzanne Corkin. HM suffered intractable epileptic seizures, often many times a day, which were poorly controlled by the drugs of the day in the 1950s. And so the decision was taken to conduct a frankly experimental surgery done by William Scoville, which was bilateral removal of the medial temporal lobe. In this drawing done at the time of the surgery, just the excision on the left is shown to show the structures on the right, but the actual surgery was to both sides. The epileptic seizures decreased afterwards. That was helpful. It wasn't complete cure, but it was helpful. But unfortunately, the unexpected consequence was that HM completely lost the ability to form new memories. He could hold on to information for a few seconds at a time. His short-term memory was intact, but his long-term memory had basically gone. But by careful study of HM, something new emerged, which illustrates further, as I shall go on, this idea of different networks mediating different aspects of learning and memory. Here's a drawing which is called technically in the field the Ray-Osterrath figure, and you can see that HM can make a pretty good direct copy. But asked 30 minutes later to draw this picture, he has no idea that there even was a picture. However, when taught to draw in a mirror, in which he had to keep a stylus in the little tram lines here of this star shape, you can see he goes over the lines initially here and is making a lot of errors on day one, but he gets better. And on day two, he's a few errors, but he's maintaining a better performance than at the beginning. And day three, he's doing really well. So it looks as though HM can learn to do this kind of motor skill, even though he apparently can't remember for 30 minutes. And indeed, when asked on day 4, shall we do this little game in the mirror, he said, kind of, what mirror? Because he couldn't remember it. And then the experimenters said, well, let's give it a go, let's try it. And of course, he then did it perfectly. So there's a sort of disconnect here between this acquired skill and an awareness that he actually had it, a memory that he actually had it. Now, that discovery was made by Brenda Milner, who worked at McGill University. And of course, the medical school at McGill was set up by graduates of the University of Edinburgh. And she says that to see HM had learnt the task perfectly, but with absolutely no awareness that he had done it before, was an amazing dissociation. If you want to know what was an exciting moment in my life, that was one. Now, all of this was done in the days before we actually had structural magnetic resonance imaging of this kind. I showed you an MRI image a moment ago, but of course that MRI image was taken many, many years later. And this is an MRI image which shows the hippocampus. And as the principal noted at the beginning of this lecture, I was appointed to a professorship in this university in 1994 and had to give my inaugural lecture in this very room introduced by your predecessor, Sir David Smith, one of the great botanists of the 20th century. And my mum and dad were in the audience, sitting just up in the middle here. And of course, over the years, I'd given my mum and dad lots of grounds for doubting that I actually had a brain in my head. So I'd recently been a subject in an experiment at the Massachusetts General Hospital in which I'd had my brain scanned. And so I showed this image of my brain in the lecture. And I presented it to my mum and dad. And I said, look, you know, I really do have a brain. At which point my father said, no proof that it's working, my boy. Now we have, I think, lots of different memory systems. And we all distinguish between short-term memory and long-term memory. I've given you a hint of the difference between procedural learning and more explicit memory. So let's just go into that in a little bit by thinking a little bit about short-term memory. This is the kind of active memory where you keep things in mind. And the basic kind of model that's emerging is the idea that you take information in through sensory registers, be they visual or auditory, and they can hold on to information for a little while, but only a matter of milliseconds, but sufficient to activate specific content within semantic memory, so that you have visual information coming in, and then there's some translation happening in associative structures in the cortex, which convert that information into being not just something that's sort of tan-colored, but it's a table, or not something with just sort of things sticking out, but it's a chair. That allows information about this specific content to enter a device called working memory, and in working memory, it can somehow hold information in an active state. And that's quite different from episodic memory, which I shall come on to in a moment. And this model has been developed by the British experimental psychologist Alan Baddeley over many years, and he supposes that the working memory system is further subdivided into a number of different systems, a sketchpad, a central executive, and an auditory short-term store. And very elegant human experimental psychology experiments have been conducted to differentiate these different components, particularly using what's sometimes called dual task paradigms. So that, I think, is the short-term memory system. We don't know a great deal about its localization in the brain. There have been some functional imaging studies of humans which have suggested that the central executive, when it's dealing with verbal information, is doing so in the left part of the frontal lobe. The articulate rehearsal loop is part of Broca's area, and so on. So there are some hints as to the localization, but we know next to nothing about the neural network that mediates working memory. There's also been some suggestion from animal experiments that there are neurons which, once they're triggered, can keep firing until the information is used when they stop firing. And that might be something that's happened in terms of reverberatory loops, but we don't really know. And it's an area where it would be really great to learn more about how this system actually operates. But let's turn to long-term memory then. In long-term memory, we have the key difference that you don't have to hold information in mind continuously in order to have the memory. Whereas in working memory, if you distract somebody, the information's gone, like you know, for a phone number or something like that. You can hold it in memory actively, but as soon as you're distracted, you've lost it, right? But in long-term memory, things can go quiescent for long periods of time. I could ask anybody here, what were you doing last Christmas? And you could bring the picture back to mind, but it doesn't mean you've had to have that image active in your brain for the last 11 months. You somehow can change your nervous system and then reactivate the memory later. So that's the key feature, as it were, of long-term memory. Now, let's look at the implicit and procedural system, in which we have actions and intentions, habits and emotions. Now, given that it's doing a variety of different things, you might imagine that there'll be different brain areas involved, but particularly in relation to actions and habits, I think it's quite interesting that there are three major areas of the brain that mediate our ability to learn habits and actions. So the first of these is the so-called motor cortex, a region just anterior to the central sulcus of the brain. And this is an area which, when you map the body surface onto it, what you have is a very interesting principle where the number of neurons devoted to a particular part of the body is not in proportion to the size of that part of the body, but in proportion to the extent to which you have to move it in a very fine, controlled manner. So as a consequence of that, the head and trunk occupies a relatively small area, whereas the hands a much larger area. And if you take this region of tissue, which would be shown across the side here, and make a three-dimensional model, this is what, as it were, it would look like, with a huge number of neurons for controlling the hands. So that seems to be a key role for the somatosensory and motor cortex, the mediating of specific movements. But it's another matter to actually intend to make those movements, or to start to make such movements. And here, the striatum, which is located in the central part of the brain, is absolutely critical. Now here we had the advantage that we can go off and do animal experiments, particularly in the rat, and from that be able to work out the actual connectivity from the cortex into the striatal region, and also from an area called the substantia nigra, called that because the cells stain in a very black way in histological material, and these contain the dopamine that is projected up into the striatum and which is lost in Parkinson's disease. So this is the structure which is absolutely vital for mediating the intention to act, and is also a region which, when you divide between the dorsal and the more ventral parts, the top and the more bottom parts of the striatum, is part of the business of transforming actions where you have an intention in your mind about what you're going to do into a habit where things get rolled off in a more automatic way. So we can get our movements going, we can use the motor cortex to actually control which particular limbs we want to do, but then there's the question of the precise timing. And this is done by another region at the back of the brain called the cerebellum. And the cerebellum mediates the coordination and fine timing of movements. It's a remarkable structure because it contains a truly colossal number of neurons, far larger than many other parts of the brain, and a very intricate structure which has been described in exquisite detail, particularly by the great school of Hungarian neuroanatomists. And we're now able to impale individual cells in rodents to actually get fluorescent images, which you can see in the microscope, of these individual cells. And the aim is to try to work out precisely how this network does the fine timing of movements that is essential for skilled performance. Now, this microscopy is being taken to a new level, and here's a wonderful image which was submitted by Michael Hauser from University College London for the Wellcome Image Awards. It didn't win, but I think it's an example of the way in which you can, with this amazing microscopy, see the individual connections of these cerebellar neurons. Now let's turn now to the declarative system. This is the system where we think of as kind of memory. The memory for events, the memory of where things are, and our memory of facts. And it's the memory that's mediated by the medial temporal lobe, by the thalamus and the neocortex. So let's just be precise about what we mean by these different types of memory and why we might need different neural networks to mediate them. My first lectureship in the academic world was actually at the University of St Andrews. And I was there in 1984 when Sevi Ballesteros won the 1984 Open. And I was standing in the crowd. I didn't take this picture. It was actually taken by Peter Adamson, who's the great-grandson of a great Scottish photographer. But I love it as an instance of episodic memory, because he'd walked up through the Valley of Death onto the 18th green, and he'd putted that last putt and won the Open. It must have been an amazing moment. But of course, there's no trial two. This isn't like learning to ride a bike. It all has to be done in one go. And I'm sure he remembered that moment for the rest of his life. So what's distinctive, I think, about the episodic memory system is that a thing can happen just once, and the nervous system can capture that information and hold on to that information potentially for a very long time. And the French photographer Henri Cartier-Bresson talked about this as the decisive moment. But he broadened the definition a little bit to try to think of moments that capture aspects of family life, such as this little boy getting his vin ordinaire for his grandma in a Parisian street. So episodic memory then is about the events of our life. In contrast, semantic memory is the organized body of knowledge that we have of things in the world. So with respect to objects, there's the question of whether they're inanimate or animate. And if they're animate, are they mammals or they're birds? If they're birds, are they flying birds or are they flightless birds, such as a penguin? If they're flying birds, are they songbirds or they're others, and so on. And what we do is we organize our knowledge, particularly in the professional sphere, in order to keep on top of things, in order to acquire new information, such that things aren't sort of random as they are in, say, the attics of our houses. Now, this distinction between episodic and semantic is one that I think is incredibly important and to some extent reflected in the way in which the medial temporal lobe interacts with the rest of the cortex. And a very interesting dissociation between the two emerges in the case of what's known as developmental amnesia. In developmental amnesia, there's some perinatal injury that happens to a baby, and then the child grows up, and later on in life there is a risk of there being some episodic memory impairments, but astonishingly these kids seem to have relatively intact semantic memory. Now this suggests that episodic memory is not necessarily the only route into semantic memory, although I think it's the typical route in, but it does at least suggest that these two can be considered as qualitatively distinct. And in a project done by my wife, Monica Munoz, she investigated in children between the ages of 8 and 16 the potential impact in later life of surgical transposition of the great arteries soon after birth, so-called blue babies. And she did this in a project at the Institute of Child Health attached to Great Ormond Street Hospital. Now, this was a huge and comprehensive project, and this slide is not intended to capture every aspect, but she went through all of the hospital records to find the cases where this operation had been happening, and found 236 over some specific time period, and then had various exclusions which had to do with whether the children were now living abroad and so on. And she finally ends up with a sample of 40, some of whom have ventral septal defect and some not. Now, when you do that and then look at these children when they're aged 8 to 16, what you find is a considerable decrease in the size of the left hippocampus, considerable decrease in the right and the mean hippocampus, without much change in the rest of the grey matter or white matter. And these kids do show some element of episodic memory deficit without the semantic deficit. Now, it's not in these cases so severe that many of them actually were full-blown developmental amnesics. But I think it illustrates the way in which we can think of these as two rather separate aspects of memory. Nonetheless, how is the transformation between episodic and semantic memory actually achieved? We've got a memory of an individual episode which can sometimes be transformed into factual knowledge that then, stripped of the spatial and contextual information, is assimilated into existing schemas of knowledge. Now, to achieve this, events have to be placed into context. You can't just have isolated events flying around without any kind of anchors that put them together. And here the anatomy is a little bit of a clue, but also you'll see that physiology helps too. So here's the hippocampus, which is crucial for this episodic memory, sitting within the medial temporal lobe. And the hippocampus is a structure here in the middle, which receives massive input in from the cortex and sends inputs out to other cortical regions. So it's not doing the job entirely on its own. It's part of a much larger neural network in order for that to happen. And what is the hippocampus providing? Well, it may in part be processing events, but on top of that, it's also processing information about space. And I'm very honored that John O'Keefe is here tonight, because he made the important discovery that if you put recording electrodes into the hippocampus of freely moving rats, you could see what he called place cells, i.e. some kind of measure of where the animal was actually located. And I'd like to try and illustrate that to you, if I may, in order to try to bring the concept alive. So let's just go into Adobe Sketch for a moment and see if we can, just a moment, let me get my glasses on so I can see what I'm doing. Let's delete that one. Right, so what we're going to do is we're going to try and record for some place cells in a rat moving around a little environment. So first of all, what we've got to do is to build our environment, and we'll do that. So let's say it's a sort of square environment like that. And then we need a rat, so let's get ourselves a rat. So let's see, make that a little bit bigger. Yeah, there we go. So some nice self-respecting rat. Give him some whiskers. Okay, and rats have red eyes, so we better give him some red eyes. Okay, so let's do that. There we go. We got ourselves a rat. Now, this rat's just going to run around this enclosure, not do much more than that, and he just sort of moves around like this and sort of sniffs things out, and then eventually gets very bored and goes and curls up in a corner. Now, John's come in with his electrode into the hippocampus, and he's going to try and record from some cells. So what he finds is that when he records from one particular cell, that cell only fires in this particular part of the world. But if he now moves the electrode to record for some different cells, then what happens is that another cell will be recording from a different part of space. And we can play this game to our heart's content, and he might find another one which he's called a new cell called the border cell, which defines the border with the enclosure. So you can pick up different cells that correspond to different parts of space here. Now, that's good, but let's now consider a slightly different situation. I don't want to do that. Come on. Now, we now build our enclosure again, and two other people, Edvard Moser and May-Britt Moser, who I was privileged to have as postdocs here in Edinburgh, who now work in Norway, also did exactly the same experiment, but this time they recorded from electrodes not in the hippocampus, but in the entorhinal cortex. And so we'll make them red again so it's nice and easy to see. And, oh no, we've got to have our rat move around a bit. Okay, so he's moving around like so, all over the place. And now what we're going to do is record from cells in the entorhinal cortex. And what Edvard Moser and May-Britt found was that the cells would clump in a way like this, and they would form a kind of grid pattern. Now, the entorhinal cortex is just one synapse back from the hippocampus. So the picture was being developed that what you had here is some kind of metric for space in the entorhinal cortex, and the hippocampus identifying where you are. Now, if on top of that, the hippocampal structure was also able to process information about events, you might be able to bind information about events to the places where they happened, and that's obviously going to be critical for episodic memory. So that's the idea, and so what we've got then is our various different cells, our place cells, where you have a hotspot for a particular bond. Here's our grid cells. There's another class of cells which are studied intensively here in Edinburgh, the head direction cells, which fire for particular angles and directions that the head is pointing. Now, inspired by this, I developed a system for studying spatial memory called the water maze, because I wanted to try and test John O'Keefe's idea that the place cells were critical for spatial memory. And the water maze is really just a large pool of water, not much more than that, with a platform hidden underneath the water, and the animal gets to swim around to try to find this platform. And I hope we're not in the presentation. Let's get into the presentation. Okay. So we have a little film of a rat doing this, and here's the laboratory as it exists in Edinburgh, and my assistant's putting the animal into the experiment, swimming through the water, which has been made cloudy with a little bit of powdered milk. And so the animal can't see this platform, it can't hear the platform, it can't feel it until it's got there. But perhaps using this spatial system, the metric of the grid cells and the place cells to know where you are, the animal can actually anchor himself in space as events happen. And I hope this film is convincing that when you now run this trial where the animal's given 60 seconds to try to find this platform which has been pushed down to the bottom of the pool, he displays a remarkable amount of spatial knowledge about where things should be, where safety should be in this particular environment. Now the platform's held down by electromagnets, and after 60 seconds we bring it up to within 1 centimeter of the water surface, so that now when the animal persists in finding it, he can climb out of the water again. So that's good. I should mention a little story. When I began in St Andrews, where I developed the water maze, I didn't have a research grant, but my wonderful professor, Malcolm Jeeves, gave me a little bit of money to get going. But that didn't last very long. And although I'd built the apparatus and the animal costs at that time were covered by the university, I still needed, bits and pieces, computer equipment and so on. So every penny counted. But there was a company, I think it was called St Ivel, if I remember correctly, which made powdered milk. And I was very young at the time and very naive. I probably still am. But anyway, I wrote a sort of pleading letter to the company saying, I'm doing incredibly important research, medical research at the University of St Andrews, and I need lots and lots of powdered milk. And would you be so kind as to provide me with a lot of powdered milk to do my experiments? And then I added in the letter, I said, look, if I eventually publish a paper on this, I'll be sure to acknowledge the company in the paper. And they wrote back saying, we'd be delighted to give you lots of powdered milk, which duly arrived in a big carton, but they said, please don't mention the company in your paper. True story. Now, I've arrived in my intellectual argument at the point where I've got this structure in the brain which is somehow processing events, but that structure in the brain also seems to provide some kind of spatial coordinate system where you keep track of where you are. So would you not want to have some system to tie these two together in some kind of way? Now this is where Edinburgh comes back into the story with David Wilshaw and Christopher Longard-Higgins, who at the time were developing the idea of distributed associative memory systems, and they and others pointed out that the internal structure of the hippocampus was very similar to what you would think of as desirable in a distributed associative memory system. So you've got the various different subparts of the hippocampus, the dentate gyrus, CA3 and CA1, which have slightly different neural networks, but in all cases, the actual synaptic connections here have to display plasticity in order for this binding process to actually happen. The question is, did that plasticity actually exist? Well, this is where my colleagues in the Brain Prize this year made, I think, some very important discoveries. Tim Bliss made the discovery that if you stimulated these synapses in the hippocampus and recorded for long periods of time, you could get nice stable potentials, which here is shown as a negativity because you're recording outside the cells. But that if you gave brief bursts of high-frequency stimulation, these potentials would get much larger. And you can see in the second pathway, they're getting much larger, and this effect is lasting for a long period of time. Now that, I think, was a very important discovery because it suggested there was the possibility in this hippocampus structure that we had plasticity of the strength of the synaptic connections. Now on top of that, Graham Collingridge, who was associated with the pharmacology department of the University of Bristol, which was developing amazing new ligands for looking at glutamate receptors, one of which was called APV, they discovered that this drug AP5, if it was put onto these synapses, at the time you gave this brief burst of high-frequency stimulation, you saw no change in efficacy. And this drug acts on a particular receptor called the NMDA receptor, which is crucial for actually changing the strength of connections. So that, I think, was another very important discovery in the process. Now, this phenomenon of long-term potentiation, the ability of synapses to change in strength when stimulated in a particular way, was touted as something that might be involved in memory. And the reason it might be is, first of all, it persists over time, and any memory mechanism must outlast the conditions of its induction. So that's a vital first property. Second, it showed synaptic input specificity. That came in part from the NMDA receptor, but what it suggested was that the storage capacity would be proportional to the number of synapses, not just the number of cells. Now, that doesn't preclude the possibility of cellular memory, but synaptic memory is probably 2 or even 3 orders of magnitude greater in terms of storage capacity. And last, there's the business of associativity, this business of putting things together, which I emphasize, and which is also mediated by this NMDA receptor mechanism. So given those properties, I became very interested in the idea of moving from the in vitro experiments that Graham Collingridge was doing by asking whether we could use this drug APV or AP5, which is acting at particular antagonist binding sites on the NMDA receptor, to see what would happen if we did such experiments in living animals. And we used our water maze, and what we found, to our great delight, was that when we gave this drug AP5, the animals were quite unable to learn precisely where the platform was located, and they would swim all over the place. They could nonetheless learn a procedural task, just in the way that HM had learned mirror drawing. So they weren't completely incapable of learning, but they were unable to learn this spatial arrangement. And this all happened at a dose of the drug which blocked LTP. Now, that was an exciting time for me, and it led me to Edinburgh because I realized then that I needed to learn much more pharmacology in order to take things much further. And so in 1986, I moved here to Edinburgh, the kind invitation of John Kelly, and he introduced me to a number of pharmacologists who enabled me to take that research forward in a much more rigorous manner. So now what we've got is a kind of circuitry in which we can actually somehow encode events, although that's, I think, perhaps still some of the mysterious parts of the thing. We can also record locations, and in that way we can tie events to places where they happen. Now, the last part of this talk that I'd like to come on to is memory retention and the updating of memory as we draw things to a close. One thing I've become very interested in is the fact that in situations of extreme novelty, you have arousal, and that helps in the encoding of memory, but it also helps in the consolidation of memory. And various models have been proposed by academic colleagues which suggest that a particular transmitter, dopamine, feeds back into the hippocampus, and when that dopamine is released in the hippocampus, you actually kind of fix memories over time. So the idea here is that you can make a memory, but that's no guarantee that it'll last. We all know that we're forgetful. But if you want something to really last, then you have to sort of drive this dopamine system as well. Now, it's interesting that the phenomenon of flashbulb memory is one that we're all aware of. My mum always remembered VE Day as a very happy day. She was a young lady at the time, 1945. I'm just old enough to remember the assassination of President Kennedy, though not very well. The fall of the Berlin Wall, there's 9/11, and then this year the election of a uniquely unqualified property boom. Now, along with the horror and surprise of the event in question, we also remember many trivial events of those days, events that ordinarily would be forgotten. So novelty arouses our attention and kind of aids memory, and we've developed a theory which explains this. And what we've got is is this film going to go? No. Let's just see if it will. Is it going to run? No, it's not going to run. Doesn't matter. It's going, is it? I'm being impatient, am I?
Just stop.
Let's go back a bit. Let's try now.
There we are.
Yep, okay, sorry, I'm being impatient. Okay, so the rat's in a little box here, and he's coming now into this dry land arena. He's not in a water maze, and he's been told that food is here today. It varies its location from day to day. He runs out, he checks that one, not there, finds the food there, digs for a while, digs up the food, and like any self-respecting rat, takes it home to eat. Now, the value of this task is that it's one that, oops, I'm getting stuck here. Oh, I know what it is. I've come out of the project thing. There we go. Not used to my iPad here. So we've got actually a series of 49 holes that we can bury the food in. And on this particular day, it might be in row 2, column 2, but on another day in a different position. And what we can do is test their memory of how well they can remember where the food is, either after a short interval, like 30 minutes, which is still within the domain of long-term memory, or after 24 hours with a test done later. The next day, say on Tuesday, we move the thing down to here, on Wednesday over here, and Thursday over here, and you can carry on this training for days, weeks, and even longer. So it's a very flexible kind of task. And what you find is that the animals hold on to the information for 30 minutes, but they've lost it after 24 hours. But if you now give these animals a novel experience of just putting them in a box with interchangeable floor surfaces, here just with paper, 30 minutes after memory encoding, so this is a kind of arousal which we think is upregulating the availability of plasticity proteins which help to stabilize memory, then what we find is that if we do that, here's the box that I've just shown you a picture of, the animal going in 30 minutes after encoding. Then, whereas typically he would forget the information within 24 hours, this novel experience later makes that thing memorable. So this is our kind of little animal model of kind of 9/11, as it were, and was published a few years ago. Now, in this series of experiments, we went on to show that this novelty was sensitive to inhibition of protein synthesis. It was sensitive to drugs that affected the dopamine system. So it was all consistent with the kind of model that people were suggesting of neuromodulation of the memory system to make things last. But we didn't have any real idea of the particular cells that would actually do that. And we wanted to get to the heart of the neural mechanism, the neural circuit that was doing that. So what we did was we decided to switch the experiment to use genetically modified mice, to do exactly the same experiment, but now in genetically modified mice. And the reason we wanted to make that change is because we wanted to use the exciting new technique called optogenetics. Now, I should explain what optogenetics is. At the back of your retina, you have rods and cones which convert light energy into electrical impulses, which then go into your brain up to the visual cortex. And one of them, I'm sure you know, is called rhodopsin, which is in the rods. Now, the opsins are the things that convert light energy into the electrical impulses. And some German biochemists in Munich some years ago discovered a number of rather strange opsins in unusual algae, which were capable of activating sodium channels. And a group in Stanford saw the publication that they'd made and suggested that it might be possible to do this in any neuron in the brain, not just in the eye, but anywhere. And they proceeded to do this by creating viruses which contained this thing called channel rhodopsin, and they would, with a surgical operation, put it into particular neurons in the brain and then lead light guides, which were not much bigger than a human hair, down into that tiny part of the brain, activate it with light, and then you'd activate those cells. So it's an amazingly powerful experimental technique. So we decided to try this optogenetic technique. You have to use blue light, so here's the mouse with the blue light, and with luck we'll have a little film of the mouse running around. Does he run around? No? Things getting a bit stuck here. Well, let's move on, doesn't matter. What we've got is a particular virus, which is a Cre-dependent virus for channel rhodopsin, and we are going to put this into the region of the locus coeruleus, which is the arousal part of the brain. But the reason to switch from rats to mice is because we were able to get a line of genetically modified mice which only express this thing called Cre in tyrosine hydroxylase positive cells. Now, tyrosine hydroxylase positive cells are cells that are for catecholamines with a noradrenaline or dopamine. And so if you put a Cre-dependent channel rhodopsin into a Th-Cre mouse, what will happen is that only the catecholaminergic neurons in that region will express the channel rhodopsin, and when you then lead the blue light down, only those neurons will fire. And that gives you a much more definitive experiment than you would just by electrical stimulation. And while I won't take you through all of the results, what we find is that we can substitute completely the behavioral novelty by just this activation of LC. And we do it with a pattern of stimulation which is playing back the pattern those cells make when the animal's moving around a novel environment. And we think that that's quite an exciting discovery and begin to give us an idea of what makes memories last any length of time. Now, there are much more to this story. There's the whole business of how information initially encoded in hippocampus becomes stabilized in cortex, how information is assimilated into the semantic memory system, and that's a big and difficult challenge for the future. But let me just go on to one or two other themes in neuroscience research which I haven't had time to cover, but which I think I should pay some attention to. One is sleep. Sleep is incredibly important for memory consolidation, and we are now beginning to understand some of the electrical patterns that occur during sleep that aid this process of memory consolidation. Another is the fact that memory can be inaccurate, and a very important thing is the unreliability of memory and the so-called misinformation effect, which was discovered by the American experimental psychologist Beth Loftus. And experiments that she and her colleagues have done over a number of years have revealed that leading questions of the kind that might be used in a court of law can actually alter people's memory of a particular experience. So if you say, "Did you see the stoplight?" that has a bigger and longer lasting effect on changing memory than "Did you see a stoplight?" It's a really astonishingly subtle effect, but a strong effect. And these experiments led her on to all manner of work looking at the intersection between neuroscience and the legal system, and recently won her the John Maddox Award. You see her here with Phil Campbell, the editor of Nature, because she faced all manner of difficulties when dealing with things like memory, apparent memory of sexual experiences that young children were said to have had when in fact they didn't have it. These being memories that had actually been sort of, as it were, imparted into them, but not necessarily of true experiences. So I think this is perhaps an inevitable feature of the reconstructive nature of memory, but it reflects an updating process that we now call reconsolidation. Another topic which I haven't covered and which I'd love to do is the whole issue of how memory decays in Alzheimer's disease. But as some of you may know, I am giving the Alzheimer's Scotland Christmas lecture next week on Wednesday, and I'll go into that research at that point. So let me just, in the last 5 minutes, touch a little bit on the future of neuroscience research on memory. How are we going to kind of take things forward to understand some of the problems in greater detail? Well, I'd like to mention here a personal experience because I think it's very important. And that has to do with the creation of the Sainsbury Wellcome Centre at University College London. About 10 years ago, I had the great privilege of being seconded to work for the Wellcome Trust in London during a governor's dividend release period, and was asked by the then director, Mark Walpert, to think about big projects that the Wellcome Trust might be interested in doing. Now, the Trust is very famous for the Sanger Centre and the sequencing of the human genome. It's also very famous for its work on human brain imaging with a major centre at UCL. But what I put to Mark was that it's that middle ground, these neural circuits in the middle that we know not very much about. And we really do need a focused effort to look at neural circuits. Now, unknown to me, the Gatsby Charitable Foundation, which is founded by Lord Sainsbury, the one-time science minister in Tony Blair's government, had also been thinking along the same lines. And so the Wellcome Trust and Gatsby got together, won each other's confidence, and decided to embark on a project which eventually went to UCL and is now called the Sainsbury Wellcome Centre. Now, as part of that, I, Sarah Caddick, who's Lord Sainsbury's advisor, John O'Keefe in the audience, and others, had to interview potential architects for this project. And it was an extraordinary day for me because I wasn't at all sure how one judges what's a suitable architect for a project like this. There were 140 applications, and we narrowed it down to 3 that were eventually interviewed. One came along with the completed building in a model. Very impressive, wonderful model. And Susie Sainsbury, who was chairing the meeting, just dismissed him, sent him out of the room, barely without a question. Because she felt, quite rightly, that you can't possibly design a building until you've talked to the scientists. The third architect who came in, Ian Ritchie, was asked the question, what will the building look like? And he said, I don't know, haven't a clue. That wasn't a great beginning for getting an architect. He said, I need to talk to the scientists. I need to go and sit in their labs and find out what's really going on there. And only then will I have a chance to really understand. Well, what's emerged is this wonderful building near Tottenham Court Road called the Sainsbury Welcome Centre. And it's now opened, and the labs are in the process of being filled. And it's a centre which is one of the first buildings ever designed to incorporate the latest scientific knowledge into its fabric, and in an open collaboration with prospective neuroscience users and the architect. And one of the PIs calls it a shockingly good building, and it's won major awards. What we did with the architect, Ian Ritchie, was we took him, we had the privilege of enough money to do this, we took him to the Salk Institute in Southern California. Louis Kahn was the inventor of the interstitial floors, and we saw those and saw how those gave you amazing flexibility for a building. Took him to Mission Bay in San Francisco, took him to Harvard. We brought him to the Informatics Center here in Edinburgh, which is a wonderful building. And out of that, and also his architects visiting UCL and other universities where they sat in on lab meetings and had days in the life, what emerged was a building which provides 2-level atria where 3 different groups can interact in a very constructive way, and it's a truly lovely building which catches the evening light given its position in the streets. So if I could, I'd just like to show you a brief clip of Ian Ritchie discussing his vision of the SWC.
The Sainsbury Welcome Centre is unique, probably at 2 fundamental levels. One is the co-location of theory with practice, experiment, coupled with the fact that you had the Gatsby Charitable Foundation and the Wellcome Trust, and coming together to create an institute that is only interested in bringing into the building the very best young minds in the world.
Right from the beginning, and I have to say this is a tremendous aspect of Ian Ritchie and his team, we as neuroscientists tried to learn a bit of architecture and what the constraints were in terms of what they could actually build. And they, on the other hand, tried to actually learn enough neuroscience so that they would be able to essentially satisfy our needs for building a building which we would be comfortable with.
I'm realizing more and more that the building is designed to sort of mix this entire domain of neuroscience, from people coming up with ideas about what brains might be to people who have data about what brains are doing, and create a new kind of thing that we're in the process of trying to define. And I think that's what's exciting about the SWC to me, is that it feels possible here.
In the world of neuroscience, you have electrophysiology, you have molecular, you have theory, you have other forms. All of that is possible here. Nothing is prescribed, nothing is out of bounds. The building is fundamentally adaptable spatially. It has verticality within the labs, and it has a plug-and-play system of services which are flexible and can allow the scientists to design their own labs with relative freedom and relatively low cost.
So we've come a long way from the barn owls, and the argument I've tried to make to you is that in order to understand some of the problems of representation in the brain, particularly in relation to memory, neural circuits matter. We do, on the one hand, need to study people in brain scanners and look at the whole system working together, and we need to work at the other end of the spectrum to look at genes and molecules. But in order to bridge these two, all too often people try to go directly from the gene right up to the top. But of course, what you need to do to make that connection secure intellectually is to understand synapses, how they operate inside neurons, how neurons work inside networks, and a full understanding of the way in which something like memory works will involve that. So that's my central argument. And so I'd just like to end with just a few words of thanks. I have a debt to so many people over the 30 years I've had the great privilege to work here, particularly to my immediate colleagues in the Center for Cognitive and Neural Systems, also to the many people who've worked in my own particular laboratory on spatial and episodic memory, on LTP, on aspects of memory loss, and particularly John O'Keefe, Maybrit Moser, and Edvard Moser, who together won the 2014 Nobel Prize in Physiology or Medicine. Not least to my Brain Prize colleagues, Tim Bliss and Graham Collingridge, here photographed with Tarje Lomo, who we were very sad didn't share the prize with us, but he came to an event that we had in London recently, so we thought we'd have our photograph taken underneath Charles Darwin. Why not? But last, I want to thank Jane Haley. Jane Haley is the lady who holds Edinburgh Neuroscience together. And all of us who are in any way involved in Edinburgh Neuroscience community know how much she does to alert us to grant opportunities, to arrange public events like this, to make sure that new PhD students get to meet each other, all manner of things. So my personal thanks to Jane, but I think that the best way of expressing that is to show you a letter that a little girl wrote a few years ago. So let's just have a look at that. It writes, "Dear neuroscientists, I'm writing this letter to thank you for the most enjoyable visit that you organized for us. I do appreciate the work you have done to make our visit so enjoyable, and I hope you enjoyed having us too. I found the lectures very interesting and I learned a lot from them. I also enjoyed making the plasticine brains and seeing the real brains. The optical illusions were great too. My greatest thanks goes to letting me win first prize for the What's in My Head competition. I'm very grateful. Yours sincerely. P.S. I apologize for the large amount of biscuits consumed by some pupils." Wonderful stuff. You couldn't make it up. So, may I wish you all a very memorable Christmas.